Okay, so students, we are now moving on to our next lecture about cell biology. And today we're going to be taking a detailed look at cell organelles. So I hope you remember from last time we talked about cell theory, and we saw that there are three parts of cell theory. All organisms are composed of one or more cells. The cell is the basic unit of structure and organization in organisms, and all cells come from pre-existing cells. So today we're going to be concentrating on this second point. The cell is the basic unit of structure and organization in organisms. So we're going to be looking in detail at how the cell is organized, exactly what we mean when we talk about organization in cells, exactly what we mean when we talk about cell organelles. So again, this is what we looked at last time. We saw that there are two basic types of cell, eukaryote and prokaryote, and the defining characteristic that distinguishes between eukaryotic and prokaryotic cells is whether or not they have a membrane enclosed, a membrane bound nucleus. Eukaryotic cells have a nucleus. Prokaryotic cells do not have a nucleus. So today we're going to be looking at the organelles of eukaryotic cells. We'll start looking mostly at animal cells. We'll say a few words about plant cells. So 2 points I want you to remember is that not all eukaryotic cells will have all the organelles that we're going to be talking about, and they're not going to have them all in the same quantity. For instance, some cells have just a few mitochondria, other cells have many, many mitochondria. And the presence or absence of organelles in a cell is related to the function of that cell. So I hope that you already know that a mitochondrion is the cell organelle connected to producing energy. So cells whose function is to produce a lot of energy will have a lot of mitochondria. Cells whose function is something else, not to produce energy, will have enough mitochondria to provide energy for the cell's own use, but not enough to export that energy in the forms of other molecules. Okay, so the presence or absence of organelles is related to the function of the cell. And this is a big theme that we're going to be coming back to again and again: structure and function. The structure of a cell is related to the function of a cell. So we can make a lot of good guesses about what the function of a particular type of cell is by looking at its structure. Okay, so here is basic cell structure. This is what we saw last time with all the different organelles labeled. I hope that you already have this sort of picture in your mind. And the definition we saw last time of just what we mean by the word organelle is a subcellular compartment or large macromolecular complex, often membrane enclosed, but has a distinct structure, composition, and function. Okay, so that's what we mean by an organelle. So the nucleus is an organelle. It's got a membrane around it. The cytoskeleton is an organelle and does not have a membrane around it. The endoplasmic reticulum is an organelle and is made of membrane. Okay, so again, the structure of an organelle is related to the function of the organelle. So you'll see, for instance, that mitochondria have a structure with a lot of folds of membrane on the inside. And because the function of a mitochondrion is to produce energy, which it does by transporting across membranes, it has a lot of membrane to allow it to carry out that function. So structure and function very closely related. Okay, pause for questions. I hope you remember how to ask questions on the forum on the LMS. Okay, so the next thing I'm going to show you is a video. It's called a video tour of a cell and just gives you a brief introduction to each of the organelles and helps you to see how they fit together to make up the whole cell. So we'll watch this and then I'll come back and talk individually about each of the different organelles.
Let's examine a cell more closely. There are two general classifications of cells, prokaryotic cells and eukaryotic cells. Prokaryotes are single-celled organisms and include bacteria and some algae. On the other hand, eukaryotic cells make up more complex organisms like plants and animals. Prokaryotic cells lack a membrane-bound nucleus, so the DNA, which is usually a single circular molecule, floats in the cytoplasm of the cell. Eukaryotic cells contain a membrane-bound nucleus that holds all of the DNA in one part of the cell, as well as many other organelles. Let's examine a cell more closely. There are two general classifications of cells. Usually, a single circular molecule floats in the cytoplasm of the cell. Eukaryotic cells contain a membrane-bound nucleus that holds all of the DNA in one part of the cell, as well as many other organelles. Now let's look at the function of each organelle in a eukaryotic cell. To gain an understanding of the inner workings of the cell, it's helpful to think of the cell as a mini city. Just like in a city, a cell is made up of many parts that have their own functions. In the cell, these parts are called organelles, which are explained next. A plasma cell membrane is made up of a phospholipid bilayer. The bilayer surrounds the cell, protecting it, and functions as a gate that allows molecules into and out of the cell. In our city, the cell membrane would be like a large fence surrounding the city that only allows certain molecules to go in and out of the cell. The nucleus is like the Capitol building of the city because it functions as the control center of the cell and houses the genetic information or instructions for what the cell is supposed to do. The mitochondria functions as the powerhouse of the cell by producing the energy the cell needs to perform its duties. This is just like a power plant providing energy for a city to use. The energy currency of the cell is ATP. The ribosomes act as factories which translate the genetic information, or DNA, from the nucleus into usable protein molecules. Proteins do the work in the cell. The Golgi functions as the post office in the cell and packages and modifies the proteins. The endoplasmic reticulum is the highway of the cell. It forms an interconnected network of tubules and vesicles and functions to transport proteins throughout the cell. So those protein-containing vesicles would be like the trucks on the highways of our city. Many of the protein-producing ribosomes are located on the endoplasmic reticulum. The lysosome is the trash man of the cell because it digests excess or worn-out organelles and proteins by breaking them down and removing them. There are actually two types of eukaryotic cells, animal cells and plant cells. Both types of cells are very similar, but there are three distinct differences. First, plant cells have a cell wall surrounding the plasma cell membrane. Animal cells just have a cell membrane. Secondly, there is often just one large central vacuole found in plant cells, whereas animal cells have a few small ones. The vacuole stores molecules needed by a cell and also isolates material that may be harmful to the cell. The final difference is plant cells have an additional organelle called a chloroplast. The chloroplast is the site of photosynthesis in a plant cell. Photosynthesis is the process of capturing sunlight and synthesizing glucose and oxygen from carbon dioxide and water. Photosynthesis is one of the most important biochemical pathways since nearly all life on Earth depends on photosynthesis as a source of energy. Animals metabolize the glucose from a plant source to produce ATP, the energy currency of the cell. Recall that all organisms need energy to do cellular processes. Do you remember which organelle is the powerhouse that generates ATP energy? That's right, it's the mitochondria.
Okay, so I hope that you enjoyed that video. So, one of the main points I want you to remember from that video is the way that cell organelles are interconnected with one another. Physically within the cell, we'll be talking about some of those connections, but also biochemically, because very often the products of one organelle are needed for another one to carry out its function. So, for example, ATP we know is produced by mitochondria and is needed by many other parts of the cell to allow them to carry out their functions. Messenger RNA is produced in the nucleus and is needed for protein synthesis by ribosomes. And the protein produced by those ribosomes is needed structurally to make up all the different parts of the cell, and also functionally as enzymes to allow them to carry out their function. So while we are looking at each of the organelles in turn, I really want you to remember that they don't operate separately. Just as if you consider a whole body, a heart is no use if it's outside a body. It only functions correctly if it is part of the whole of the body. Okay, so we'll start with the cell membrane. So that is the membrane around the outside edge of the cell, sometimes called the plasma membrane because it surrounds the plasma or the cytoplasm. So the membrane of the cell is what forms a barrier between the inside of the cell and the outside of the cell. But it's not just a passive barrier. It doesn't just separate like a wall. It actually controls what passes in and out of the cell. So you can think of it as a wall with gates or doors in it. There are a number of different ways that things can pass in and out of the cell. They can pass by diffusion, as we'll learn soon, but they can also pass by active transport, which is being transported across the cell, needing energy, and by passive transport, which is a specific method of transport but doesn't need energy. So we describe the plasma membrane of the cell as being selectively permeable. It allows molecules to pass through it, but it selects which molecules are allowed to pass. So here is a more detailed picture of the cell membrane. You can see that there are two layers. Okay, we'll be looking in detail at exactly what makes up these two layers of the cell a little bit later. So for now, I'll just say that the cell membrane consists of a phospholipid bilayer, two layers of phospholipid molecules with a lot of other proteins involved with it. Okay, so here we can see a protein channel, which is involved in transporting other molecules through the membrane. Here we can see proteins attached on the inside of the membrane or on the outside of the membrane. We can also see carbohydrates attached onto the membrane. These are often involved with interactions with other cells outside the cell, signaling to other cells and also sticking physically to other cells, adhesion, sticking together. On the inside of the cytoplasmic membrane, we can see the cytoskeleton, which is made up of proteins and helps to give the cell its shape. So there's a layer of cytoskeleton just under the cell membrane. Okay, so we'll be looking in detail at the cell membrane in a few lectures' time. Okay, another pause for questions. Okay, so the next organelle I want to talk about is the nucleus. So we've seen pictures before of the nucleus, a very distinct organelle that can be seen quite easy with a microscope. So the function of the nucleus is to contain and to store the DNA, the instructions for making the cells, the hereditary material of the cell. The DNA is packaged into chromosomes. It's organized with proteins attached to it and stored inside the nucleus. So the nucleus controls the expression of genes in the cell. Genes are the instructions to make proteins. You'll be learning a lot more about genes and how their information can be used to make proteins next semester. So we've seen that eukaryotic cells have a nucleus, prokaryotes don't have a nucleus. So because eukaryotes have a nucleus, because they are able to contain and organize their DNA in this way, it means that gene expression, the use of genes, switching genes on or off at a particular time in a cell, is more possible to control very precisely, very finely than it is in prokaryotes. So in a eukaryote, the nucleus is important to allow the cell to carry out its functions in a very controlled and precise and exact way. Another reason for having a nucleus is that separating the genetic material from the rest of the contents of the cell helps during cell division to make sure that the two sets of genetic material are very precisely placed in the two new cells after cell division. And we'll be looking at exactly how that's done in a few weeks' time. So because of the nucleus, because of having the nucleus and because of being able to organize DNA in the nucleus, eukaryotic cells can have a larger genome. They can have more DNA, more genes, more instructions to make more different things than a prokaryote has. So the structure of a nucleus, we've seen these photos before. Here is a nucleus looked at under a light microscope. Here is a stained nucleus. So here a dye has been used that stains the DNA yellow. So we can see that the DNA is very concentrated. It's there in the nucleus and not there in the rest of the cell. The red staining here is staining the cytoskeleton of the cell. So we can see that the DNA is in the nucleus and not visibly in these cells anywhere else in the cell. So the nucleus is usually approximately spherical, although some types of cell have nuclei that are different shapes. It takes up approximately 10% of the volume of the cell, remember that there are so many different sizes and shapes and types of cell, but I'm speaking very generally here. Outside the edge of the nucleus is the nuclear membrane. So the nuclear membrane separates the inside of the nucleus from the rest of the cell, from the cytoplasm of the cell. And the nuclear membrane is actually a double membrane. That's two layers of phospholipid bilayer. So we've seen that the cell membrane is a single layer of phospholipid bilayer. The nuclear membrane has two layers of phospholipid bilayer laying against each other with a small gap in between. So surrounding the nucleus, the nuclear membrane or nuclear envelope, and this membrane is connected to the endoplasmic reticulum. It's continuous, it's attached to the endoplasmic reticulum. The inside of the nucleus is separate from the cytoplasm, but it is connected to the cytoplasm by channels through nuclear pores. So these are specific structures in and on the nuclear membrane that allow things to pass in and out of the nucleus. So inside the nucleus is chromatin, which is the word we give to DNA and the proteins that package it up. And here you see a representation of the nucleolus inside the nucleus. So look at the word carefully. Nucleus, nucleolus. Okay, there's an extra O here, nucleolus, an extra O and L. So the nucleolus is the area inside the nucleus where ribosomes are made. So it's not a separate structure as such. It's not separated from the rest of the nucleus, but it's a very condensed, very busy part of the nucleus. Okay, so the nuclear membrane, here's a close-up picture of what we were just looking at. So you can see here quite clearly the two layers. So each layer here is a phospholipid bilayer. And here are the nuclear pores. Here's one that's been cut through the middle. So you can see the way that it forms a channel through the nuclear membrane. In between the two layers of membrane is a space which is called the perinuclear space. And the outer membrane is continuous with the endoplasmic reticulum. It folds out and becomes the endoplasmic reticulum. Okay, and the other thing you can see here is ribosomes attached to the outside of the nuclear membrane, and these also are continuous, attached to the endoplasmic reticulum as well. Okay, here is an electron micrograph showing a section of nuclear membrane, and these structures that you can see are the nuclear pores. They look a little bit like little flowers or something, all packed closely together. And you can see that there's a lot of them. They're packed quite closely together on the nuclear surface. So a lot of passage in and out of the nucleus. Remember, structure and function. The structure shows lots of pores allowing passage. The function is to need to allow lots of things to pass in and out of the nucleus, but not just randomly. So the nuclear pore controls what can pass through it. It regulates the passage of large molecules, small molecules, things like water molecules and small building blocks like nucleotides or amino acids can pass through these pores quite easily, just through the hole in the center. Larger molecules like an RNA or a large complete protein can only pass if the nuclear power allows them to pass. So here's a more detailed picture again. Here are the two layers of the nuclear membrane. And here is a nuclear pore sitting in the membrane and making a hole in it, if you like. So you can see that the nuclear pore is made up of lots of different parts, given different names. Like this part looks a little bit like a basket, so it's called the nuclear basket on the inside of the membrane. There's a network of proteins on the inside of a nuclear membrane, which helps to give it a structure and help it to keep its shape. And then here on the outside, here is the ring-like structure of proteins that we can see here. Here we're looking at it sort of from the side. Here we're looking down on this structure. So there's a hole in the middle here. So this is what we can see here, and a hole in the middle of the nuclear pore that allows molecules to pass in and out through that hole. Okay, so the other thing we have inside the nucleus is DNA, or packaged up into chromosomes and chromatin. And again, this is something we're going to be looking at in more detail later. But here you can just see a diagram of the naked DNA double helix, which I hope you're familiar with. But then it starts being packaged up by wrapping it round the outside of a ball of proteins called histones, and the DNA is wrapped around the outside of that ball of histones. Then each of these structures, the histones of the DNA, are packaged close together. So you can see here the histones are organized. They're actually organized into a sort of spiral shape with the DNA wrapped around the outside of each one. So organized and compacted. It's not just randomly tangled together. It's organized in a very precise way, which is important for the function of DNA in the nucleus, because the instructions of this DNA, if you imagine the DNA as a long line of instructions, of information, of writing, a particular part has to be found and undone to make it useful. Okay, so histones packaged up onto nucleosomes, arranged into a thicker fiber. This is then coiled around again and again and again. And that is the state that DNA, that chromatin is usually found inside the nucleus. Then, if the cell is going to divide, the chromatin becomes packed even more tightly into discrete structures called chromosomes. So this is a condensed chromosome. This is what we'd see when the cell is preparing to divide. And when it's all packaged up tightly like this, the DNA becomes easier to organize, to separate, to place in two new nuclei. You can see this sort of structure when it's all lying wrapped around itself. It would be hard to take just one part from here. But when it's packaged up, it's a lot easier to organize into two new nuclei. So each chromosome, each of these structures, is a single long DNA molecule carrying lots and lots of information. Well, actually, as this is two chromosomes, sister chromatids joined at a centromere, this is actually two DNA molecules which will separate during cell division. So histone proteins are the ones which condense and organize the DNA into chromatin. So here again is the nucleolus. The nucleolus is a structure within the nucleus, but it doesn't have a membrane. If you think of the early days when people were first looking at cells in the microscope, inside the nucleus, they saw an area that was even darker than the rest of the nucleus. And that is what they called the nucleolus. And at that time, they didn't know what it was. We now know that the nucleolus is where ribosomes are being made. So ribosomes are made up of protein and of RNA. The RNA is made in the nucleus, and to make lots of ribosomes, you need to make lots of RNA. There are lots of loops of DNA, but the RNA to make the ribosomes, the ribosomal RNA, is being copied from. So this structure, the nucleolus, looks dark, not because it's different to the rest of the nucleus, but because there's lots of things packed very tightly together. I like to make an analogy with perhaps a market. If you just look at a market and the people in a market from above, usually the people are spread all over the market. But then if one person starts shouting that they're going to be selling something for a low price, everybody moves into that area and stands close to that person because there's something interesting going on. So again, if you look down from above, you would see a very condensed place, a part where there are lots and lots of people and less people in the rest of the market. So that's what the nucleolus is a bit like. There's a lot going on there, something interesting going on, so it looks very condensed. Okay, a pause for questions. Do ask any questions on the LMS. So ribosomes next. Ribosomes are found in the cytoplasm, and their function is to make protein. So information from the DNA in the nucleus is transferred to an RNA molecule by a process called transcription, which we'll look at in detail next semester. And then it moves from the nucleus into the cytoplasm. So you'll realize that it has to move through a nuclear pore. So in the cytoplasm, the messenger RNA becomes attached to ribosomes and is transcribed to make protein, is translated to make protein. So this process means that the information to make the protein can be stored in the nucleus, but it's used in the cytoplasm by the ribosomes. Okay, a transfer of information. So, as I said, we'll be looking at transcription and translation in detail next semester. Okay, so this is what a ribosome looks like. It's made up of RNA and protein, and these two things are sort of wrapped around each other to make one structure. A ribosome has two subunits, a large subunit and a small subunit, and in between the two, the messenger RNA can provide the information that's needed to make proteins. So here is the messenger RNA. Here is where a protein is being made. This is transfer RNA, you might have heard about. And this is a protein, a polypeptide, amino acids joined together in a chain to make a protein. Okay. So ribosomes are found in the cytoplasm, free in the liquid of the cytoplasm, and also attached to the rough endoplasmic reticulum. And again, you'll see why it's found in both ways when we look at translation and protein synthesis. Here is just a diagram to show how complex the ribosome structure is. This diagram shows the large subunit with dark grey RNA and brown protein. And here is the small subunit with light grey RNA and blue protein. And here is the transfer RNA in the middle. So you can see that there is a lot of RNA and a lot of different proteins. And remember last time when I was talking about discoveries, in the year 2000, the X-ray diffraction structured down to a very precise level of the ribosome was worked out. And since then it's been refined. And now we know in great detail exactly where each of these folds and parts and bits of the ribosome are. It's not at all just random. It's very highly organized to make a very efficient machine to make proteins. Okay, another pause for questions. Okay, so the next structure is the endoplasmic reticulum. Endoplasmic reticulum is described in two ways, rough and smooth endoplasmic reticulum. And the endoplasmic reticulum, or ER, has the function of manufacturing, processing and transporting many biological molecules. The difference between rough ER and smooth ER is that the rough ER is covered with ribosomes, little knobbles of ribosomes on the membrane structure of the endoplasmic reticulum. And the rough ER is involved in producing and processing proteins, especially proteins which are going to be exported from the cell. And the smooth endoplasmic reticulum is more involved in the production of lipids and carbohydrates. So here is the structure of the endoplasmic reticulum. As I said earlier, it's attached to the outer membrane of the nuclear membrane, which you can see here. And here you can see the folds of the endoplasmic reticulum. So a very folded structure. And this is the rough endoplasmic reticulum covered in little knobbles of ribosomes. The folds are called cisternae. And here, budding off from the rough endoplasmic reticulum is the smooth endoplasmic reticulum. Okay, so again, they're continuous with each other. So one big structure made of membrane, made of phospholipid bilayer, continuous with the nucleus. There's a channel here that goes into the space in between the folds of the membrane, and in the inside of where it becomes these more tubule-like structures. Okay, so there's an inside and an outside, and lots of processes are going on here which are involved in making and processing proteins and other membranes and other molecules in the cell. So another thing about being made from membrane is that membrane can change its shape. It's a fluid structure and it can change its shape and bud small parts of membrane off to make a membrane vesicle. And inside these vesicles, molecules can be transported to other parts of the cell. So endoplasmic reticulum, making, processing, transporting proteins and other important molecules in the cell. Okay. So Golgi apparatus. This is shown here in the diagram, and if you remember in the video it was described as being something like the post office because it packages and distributes the proteins and other molecules. So again, the Golgi apparatus is made of membrane, many folds of membrane, and membrane vesicles which bud off from the endoplasmic reticulum can then attach to the Golgi apparatus and let out, release their contents into the Golgi apparatus. Those contents then can pass through these many folded layers of membrane that make up the Golgi and then be passed out again in vesicles on the other side. And you'll see that one side of the Golgi apparatus here is very close to the cell membrane. So vesicles again can bud from the Golgi and attach and release through the cell membrane. So that's what we can see going on here. Here is an electron micrograph of the Golgi apparatus. So this is a real one. And here is a diagram interpreting what we can see here. So many folds of membrane with two sides to it. One side which is nearer the cell nucleus, and here transport vesicles can come in and attach to this membrane, release their contents. The contents passed through the layers of membrane to the transface, the side nearer the cell membrane, where the vesicles can be released containing the completed, finished molecules, proteins. They can then be passed very quickly to the outside membrane of the cell and released on outside the cell. So that's the sort of picture we see for a cell which is making molecules which are needed elsewhere but are being released outside. So perhaps if you're thinking about a multicellular organism like a human being, it's something that's being released into the bloodstream. Okay, another pause to think about any questions you might have on that. Mitochondria, yet another organelle made of membrane. So the function of a mitochondria is respiration. Respiration using oxygen, aerobic respiration, which is a series of biochemical reactions which produce ATP, which I hope you've already learned is a high energy molecule. It stores energy in a chemical bond in a form that's very easily released and used in other biochemical reactions in the cell. So we think of ATP as being the energy currency of the cell. So ATP is made in the mitochondria. Remember, it's a bit like the powerhouse of the cell. A powerhouse produces electricity for us to use in our homes and in our classrooms. The mitochondria produces ATP to be used by other parts of the cell. The energy is produced by a chemical reaction that takes place on both sides of a membrane. You'll be learning the details next semester in metabolism course. But to allow this reaction to take place efficiently, there has to be a lot of membrane inside the cell. So there are lots of folds of membrane called cristae or infoldings of the membrane. And the mitochondrion actually has two membranes, an outer membrane, which sort of acts to separate the inside from the rest of the cell contents, and then the inner membrane, which is the one that's so very folded, and that's the one that has the enzymes which carry out aerobic respiration. So again, here is an electron micrograph of a mitochondria, and you can see the outer membrane here and the inner membrane being folded into these folded structures inside. And here is a diagram interpreting it. So the outer membrane shown in dark brown, the inner membrane folded and folded in light brown, and a matrix inside with lots of enzymes carrying out the function of the mitochondria, the energy production. The outer membrane acts a bit like a passive sieve. It stops large molecules going into the mitochondrion. The inner membrane has a lot of selective transport going on, because that's an important part of the respiratory reactions that produce the ATP. Another point that you can't actually see on this diagram is that mitochondria actually have their own DNA, a little ring of DNA, just a relatively small amount of DNA, carrying instructions to make some of the mitochondrial proteins. And this is called a nucleoid. And it's rather more like a bacterial DNA. It's not packaged up in the same way that the DNA in the nucleus is. So you'll be learning a lot more about metabolism and respiration next semester in the metabolism course. Okay, and then we'll pause for questions. And the next structure is the lysosome. So I hope you remember from the video that that was described as being the rubbish collection, the trash collection, the waste disposal and recycling in the cell. So the function of the lysozyme is to break down foreign particles that have been brought into the cell or worn out cell compartments and break them down to produce the individual building blocks. So the lysosome will take a large protein molecule, for instance, and break it down to individual amino acids. Take a large RNA molecule and break it down to individual nucleic acids, nucleotides. So we can think of it as getting rid of waste in the cell, but also recycling, releasing them in a form that they can be reused. So the inside of a lysosome has digestive enzymes which break down biological molecules. And if these enzymes were free in the cytoplasm of a cell, well, they'd just digest the whole cell, it would break it down itself. So they're kept packaged away, stored away inside the lysosome, so they can't damage other parts of the cell. And another way that they're controlled is that the inside of a lysosome is quite acidic, about pH 4.8, and that is the pH that the lysozyme enzymes work best at. So if a lysosome is broken open, for instance, and its contents released into the cell, the pH of the main cytoplasm of the cell is about neutral, is about pH 7. So the digestive enzymes are less active at that pH than they are at pH 4.8. So that is a second way that the cell protects itself from digesting itself with its own digestive enzymes. So here is the structure of a lysosome. Here's an electron micrograph, and really they just look like sort of fairly blobby structures, fairly circular, with contents inside. Here is a diagram interpreting what we're seeing. So there's a membrane around the outside, and inside there is a mixture of lots of different enzymes, hydrolytic enzymes, enzymes for breaking down other molecules. Lysosomes are mostly quite small, although there's quite a variation. You can see here there's a large one, a medium one, and a small one. They're more or less spherical, and they're surrounded by a single layer of phospholipid bilayer. So I mentioned that the inside is quite acidic. This low pH is maintained by an ion pump, which pumps ions across the membrane and keeps the pH on the inside quite low, quite acidic. This is an energy requiring process. It needs ATP, and it actively transports hydrogen ions across the membrane to give the acidic pH inside the lysosome. Similar to a lysosome, but with a slightly different function, is a peroxisome. So these look very similar, little spherical membrane bound structures, but what's inside them is different. So peroxisomes have enzymes that can break down toxic byproducts of cell metabolism, such as hydrogen peroxide, and there's a urate oxidase core, a crystalline core inside the peroxisome. So similar to a lysosome in that it's getting rid of toxic products, but the particular things it gets rid of and the way that it does it is different in detail. Okay, another pause for questions. So the next thing I want to talk about is the cytoplasm itself. We tend to think of the cytoplasm as just being the water, if you like, in between everything else. But actually, the cytoplasm describes everything that's packed together inside a cell. It's quite thick, a thick gel of macromolecules, big molecules. So here RNA is illustrated in blue, ribosomes in green, and proteins in red. And you can see that these molecules are packed quite closely together with not much space in between. The liquid part, the aqueous part in between, is called the cytosol. But the cytoplasm itself is quite thick because there are so many things dissolved in the aqueous part inside the cell. One of the things inside the cytoplasm is the proteins of the cytoskeleton. So the cytoskeleton is rather like a skeleton inside a multicellular animal. It's something stiff that gives the shape to the cell. So there are three different types of molecules which make up the cytoskeleton. Microtubules, which are shown here in green, actin filaments, which are shown in red, and intermediate filaments, which aren't stained in this picture. The nucleus is stained in blue. So you can see that the microtubules tend to be going from the nucleus outwards, whereas the actin filaments tend to be wrapping around the cell inside the cell membrane. So together these give the cell its shape. The actin filaments supporting and shaping the outside shape of the cell, and the actin, the microtubules anchoring the nucleus and the outside of the cell in relation to each other, so holding everything in place. So here is another photograph showing the cytoskeleton during cell division, during mitosis. So here again we can see staining. The intermediate filaments here are stained in red, so a network of intermediate filaments, and microtubules here stained in green, forming the spindle of the cell. We'll think about that more in a few lectures' time when we think about cell division. And here the condensed chromosomes are shown stained in blue, and they're in the process of being pulled apart by the spindle to make two new nuclei. So the cytoskeleton important all the time for giving the cell its shape, and important during cell division for carrying out the movements and the shaping that needed to form two new cells. Similar to the cytoskeleton are the centrioles. So centrioles are also made from microtubules, but arranged in a very particular way. So arranged in 9 sets of 3. So each of these long tubes is a microtubule molecule, and 3 are arranged side by side, and 9 of them are arranged in this sort of way to produce a circle. And then each cell has 2 centrioles, a centriole pair, which are at right angles to each other. So in this electron micrograph, here is one centriole seen from the end, so it looks rather like a circle. We're looking at it end on, and the second one seen from the side, so it looks like a rectangle. So this one we're seeing sort of up against its side, so it looks like a rectangle. And the centrioles are important in allowing the spindle to form correctly during cell division. Centrioles are only found in animal cells, not in plant cells, although plant cells do manage cell division quite well, but I think they make it more efficient in animal cells. Another microtubule structure are cilia and flagella. Cilia and flagella are similar structures. The main difference is how many and how long a particular cell has. Both cilia and flagella are important in providing movement. So in microorganisms, single-celled organisms, the flagellum can move the whole cell. In multicellular organisms, cilia can make things move past the cell. So for instance, the human gut is lined with cells with many cilia on the outside, which help the movement of liquids through the gut. So cilia and flagella are made of with microtubules again arranged in a particular way, but different to the arrangement we saw in centrioles. So we can see here these sort of circles of microtubules. You can see what we're seeing here. So this is looking end on at this sort of structure. So pairs of microtubules that can slide against each other and produce movement, produce bending, produce movement in relation to each other. especially if they are attached to a motor protein. So that's a protein which can use energy to move, to change its shape and change the shape of a whole microtubule. So dynin is a motor protein. Okay, another pause for questions. So now just to say a few words about plants. We're not really going to be studying plants very much in your course, but you might need in the future to know something about plants because, for instance, plants produce biological molecules which can be important as drugs. So just a quick look at plant cells. So we've seen that plant cells outside their plasma membrane have a cell wall. And the cell wall is responsible for giving the strong structure to a plant. So, you know, if you walk outside, you can see plants, you can see grasses standing upright, you can see big plants like trees. And these are hard because of the cell walls that each cell has. So they can form a framework a bit like a skeleton for a plant. The molecule that's mostly found in plant cell walls is cellulose, a polysaccharide. You'll be learning exactly what I mean by polysaccharide in a few weeks' time in the main biochemistry lectures. Other molecules could also be present, but one important thing about cellulose is that many animals use cellulose as a food source. So grazing animals like goats or cows or elephants or any animals which eats plants, a lot of the energy they get from their food comes from the cellulose on the outside of the plant cells. You saw from the video that another feature of plant cells is a large vacuole. Animal cells sometimes have small vacuoles, which I haven't really talked about, just small membrane enclosed structures containing and storing other things. Plant cells tend to have one large vacuole that can be used to store water. And then if water is plentiful, the vacuole is very full and the plant cell is stiff and stands upright. But if there is less water, the water inside the vacuole is used up, the plant becomes less stiff and droops. And if you've ever kept a plant inside your house and forgotten to give it any water, you'll know that the first thing it does is go floppy. And that's because its vacuole is emptying out its water and the cell becomes less stiff. So the vacuole can also be involved in breakdown and recycling cell components in plants, a bit like lysosomes, and it can also be used to store other nutrients as well as water that are needed by the plant cells. So the third structure in plants is chloroplasts. So again, these are structures which only plants have, and these are the structures which carry out photosynthesis using energy from sunlight to synthesize ATP and NADPH, which are energy-rich molecules and which can then be used to make other molecules. So I mentioned the cellulose on the outside of the plant cell being important as a food source for animals, and eventually for us. We eat some plants directly, other times we eat the meat from animals who have eaten plants. So photosynthesis is used to make many molecules, but including cellulose, which is an important food source. Photosynthesis has two stages: the light reaction, where energy from the sun is trapped in the pigment molecules, often chlorophyll if it's a green plant, and then the dark reaction, which doesn't need light, but the energy from the light reaction is used to synthesize sugars. Again, you'll be looking at more details of that process later. So here is just a diagram of a chloroplast. Again, two layers of membrane on the outside of a chloroplast, and then a third membrane inside called a thylakoid membrane. And the pigments, the chlorophyll or other pigments that trap the light, are found in the thylakoid membrane, whereas the dark reaction, the other reactions to produce carbohydrates, takes place in the liquid part inside the cell, the stroma. And another point about chloroplast is that, like mitochondria, they have their own DNA, a little ring of DNA carrying the instructions to synthesize some of the proteins that are needed by the chloroplast. Okay, so here's just a diagram showing chloroplast inside a plant cell. You can see that each cell has lots of chloroplasts, lots of energy production, lots of light trapping going on inside green plants, providing energy to make carbohydrates like cellulose, which then can be food for other animals. And here's an electron micrograph of a chloroplast. Again, you can see the membranes inside containing the pigments, the chlorophyll. And you can also see that the chloroplast is near the outer layer of the cell, sort of where it will receive most light coming in through the membrane. Okay, any questions on that? Okay, so I just wanted to say a few words about some terms that I've used that might seem confusing. There are a number of words that I've used which are similar and which I find that many students get mixed up. So nucleus, I hope that you're all familiar with the nucleus. That is the membrane-bound compartment inside the cell which contains DNA. But the nucleolus, so just same word but with an OL here, nucleolus, is the dense area inside the nucleus where ribosomes are made. And the nucleosome is the structure which packages up DNA by wrapping the DNA around histones. Each of these little balls of histone and DNA around the outside is called a nucleosome. It's the first level of organization of DNA. So three similar words which have a different meaning. So if you're in an exam and the question asks you about the nucleolus, please do not tell me about the nucleus. If the question asks about the nucleosome, please don't tell me about the nucleus. Okay, make sure you know each of those words and that you look carefully to see which one is actually being used. And again, centrosome, centriole, and centromere, similar words, and all important in a similar process. So we'll look at these again when we look at cell division. But the centrosome is the cell organelle which acts as a microtubule organizing center. So this area where microtubules are organized is called the centrosome. And in the centrosome is a pair of centrioles. Okay, so that is the microtubule structure that we see. And then we didn't talk about this today, but we will do. The centromere is the part of the chromosome that the spindle fiber attaches to pull them apart during cell division. So centrosome, centriole, and centromere, again, three similar words with slightly different meanings. Please try not to become confused. And that's just a picture of the whole cell to remind us as we finish, but we're not just thinking about each of these structures by itself, but they are all packed together inside one cell, all interacting together and important with each other. So here's a cell, you can see the nucleus, around it the nuclear membrane, the endoplasmic reticulum attached to the nuclear membrane, lots of ribosomes on the rough ER and smooth ER, the Golgi apparatus, vesicles being passed around, microtubules making up the cytoskeleton, mitochondria, lysosomes and peroxisomes. Here's lots of cilia on the outside of this cell, nuclear pores, the plasma membrane, the nucleolus within the nucleus, the nuclear envelope of nuclear membrane, chromatin inside the nucleus, just showing how everything is packed together inside the cell. Okay, so just to remind you, please do go to the textbooks and read. This is the textbook I've taken some illustrations from. Leninger, older copies in the library where you'll look at chapter 2, or more recent editions. Here's the 5th edition, which some of you might have a soft copy, or the 7th edition, chapter 1, section 1 of the Cellular Foundations of Biochemistry. So please do look at that. And that's all I've got for you in this lecture. So see you next time.